welcome back to our exploration of the ICH GCPs, and we are on session 5. Let's start with a multi-center trial definition. It is a clinical trial conducted according to a single protocol, but at more than one site and therefore carried out by more than one investigator. Site here refers to a clinical research site, so multi-center is a protocol that is at several clinical research sites. This could be in one country, several sites, or this could be globally at several locations. A non-clinical study, this is a biomedical study that is not performed on human subjects. So in clinical trials, we do have studies that are not conducted on human subjects, and there are many varieties where in some situations it's just data that is actually being looked at, and certain algorithms are run to see the potential for additional studies or to see what that data brings, or they're just looking at trends for subjects over a specific period of time. It could be in the past, it could be in a current situation. So those are non-clinical studies. Opinion, and in this case, this refers to the opinion of the IEC, the Independent Ethics Committee, or the IRB, and is a judgment and or the advice provided by an IEC. Again, this could be one that is also provided by an IRB, and it relates to the documents that are submitted to start up a clinical trial. It relates to documents that are submitted periodically to the IEC as well. So whatever judgments or advice that they have are considered to be an opinion in clinical trials. Let's talk about a protocol. If you are in clinical research, this is the Bible, right? This is what we use to conduct clinical trials, and it is a document that describes the objectives, design, methodology, statistical considerations, and organization of a trial. The protocol usually also gives the background and the rationale for the trial, but these could be provided in other protocol reference documents. Throughout this ICH GCP guidance, the term protocol refers to protocol and protocol amendments. So that's the protocol, and that's its definition. It could be anywhere from 1 page to over 300 pages, depending on where it is. Now, in many cases, what sponsors are doing is they have the protocol written with all the methodology in it, and then they would have and they have different names for it. It could be a protocol extension document. It could be a protocol guidance document. It could be an operations manual. So what they do is everything is in the protocol, and then the operational piece as to how things will actually roll out, what's going to happen per procedure, and all the operational things around that now would go in a separate document. So there's a lot of that happening in the clinical research world right now, but just be aware that the protocol captures everything, and all the other documents related to a clinical trial are formed from what is in the protocol. So this is what gives you guidance for the study as well. So protocol amendment is just a written description of changes to a formal clarification of a protocol. Now, there are consequences to a protocol amendment. For administrative changes, those are really simple. You might just get a notification about it. Maybe they're changing the grammar or they're switching around some paragraphs in the protocol. But the more in-depth the amendment is, and the more it affects the changes, it affects the scope and the breadth of the study, will determine how it's going to be moved forward operationally. So if there is a protocol amendment that completely changes the way that the investigational product is going to be administered, there are quite a few consequences of that. Usually the protocol will have to go through certain processes, reapproved again. Sites have to submit it again to their IRB. Site staff have to be trained. Patients have to be notified. It might affect the informed consent. It might affect the revision of several documents. So just always be aware whenever you hear about a protocol amendment. Just want to make sure that what type of amendment it is so that you can understand how it actually affects you as you're running clinical trials. Quality assurance we had discussed earlier, and these are all those planned and systemic actions that are established to ensure that the trial is performed and the data are generated, documented, and reported in compliance with GCP and applicable regulatory requirements. So quality assurance is a system of actions. It could be audits, it could be all the periodic monitoring, it could be central monitoring. So all of that, those tools and processes that are in place to ensure that the trial is being performed in compliance. That's what QA is. Then quality control are the actual operational techniques and activities undertaken within the quality assurance system to verify that the requirements for the quality of the trial related activities have been fulfilled. So these are actually the operational techniques, the actual activities that are going on to ensure that these the trial related activities have been fulfilled. So quality assurance captures everything. It is the map, and quality control is actually what you're going to be doing, those different techniques that you're going to undertake to ensure that everything is implemented accordingly. Let's talk about randomization, a big term in clinical trials as well. It is the process of assigning trial subjects to treatment or control groups using an element of chance to determine the assignments in order to reduce bias. So randomization now in modern clinical trials typically is done through a database or a system that it assigns. It assigns the scheme to patients. So say that you are conducting a trial that has a placebo in it. It is this system that would say, for the first subject, they're going to be on the investigational product. The second subject might have placebo. There's so many different types of randomization schemes. But now, again, it's not something that is done. In the past, we used to do it with maybe an Excel spreadsheet, an envelope. Each patient, we assign them to different groups. We'll say, this person is going to be in group A, this person is going to be in group B. Now there's something called an interactive web response system, and it's web-based, and it's a system where that manages all the randomization assignments as well as the investigational products. So that's very common across many clinical trials. But in some cases, I'm sure that if that system cannot be used, there are other ways of actually setting up randomization in the trial. Regulatory authorities are bodies having the power to regulate. In the ICHP guidance, the expression regulatory authorities includes the authorities that review submitted clinical data and those that conduct inspection. These bodies are sometimes referred to as competent authorities. We all know who they are. Per country, in the US is the FDA. In the European Union, it's the EMA, and then every country, some countries are like in Canada, you have the Minister of Health and its offshoots. Many other South American countries is the Ministry of Health. And so those are the authorities typically at the national level that regulate clinical trials. Serious adverse event or serious adverse drug reaction, SAE or SADR. We had talked about what adverse events are. Now, SAEs or SADRs are huge in clinical trials. And if there is anything that you want to pay attention to, are these two terms. So an SAE is an untoward medical occurrence that that can happen at any dose, right? It can result in death, is life-threatening, requires inpatient hospitalization or prolonged of existing hospitalization, results in persistent or significant disability, incapacity, or is a congenital anomaly or birth defect. Okay? So I just want to clarify results in death, which means that the event resulted in death. So a lot of times there's confusion between, a patient had a car accident and then they died as a result of the car accident. So the SAE actually is the car accident, is not death, right? So just remember that it's what the event that causes these things to happen versus these things that happen, right? So it's life-threatening, maybe they were in a situation where they had a heart attack, and it was, I mean, it caused them to go to the hospital, for a number of days. So it's basically that event that caused them to be hospitalized, and it was life-threatening. And in some cases, one event can result in all of these. So that's something you want to pay attention to. Oh, and let me go back to source. Let me go back to a serious adverse drug reaction. So it's pretty much captured in the same way. If anything happens and these events are a result of that, then they're still recorded as an SAE. Source data is all information in original records and certified copies of original records of clinic findings, observations, or other activities in a clinical trial necessary for the reconstruction and evaluation of a trial. Source data are contained in source documents, original records, or certified copies. Source data is extremely important if you are a clinical researcher, because it's the first point in which you capture information. So in the clinical research protocol, let's say you have to draw blood at certain periods. Wherever you capture that information, that's going to be the first point. That's your source data. If you have to do blood pressure, whatever procedure you're doing, and the capturing of that data, the first place where you capture it, that's going to be your source data. And I believe I mentioned this in the previous recording. If you capture it on a piece of toilet paper, that's where source data is. So because that will show you the accurate picture of the data that is actually being captured. Then you have source documents. So source documents are original documents, data, and records, e.g. hospital records, clinic and office charts, laboratory notes, memoranda, subject diaries or evaluation checklists, pharmacy dispensing records, recorded data from automated instruments, copies of transcriptions, certified after verification as being accurate copies, microfiche, photographic negatives, microfilm or magnetic media, X-ray, subject files, and records kept at the pharmacy, at the laboratories, and at medical technical departments involved in the clinical trial. So this is self-explanatory. You can read it over and over. Those are what source documents are. So source data is the data that comes from the capturing of this. Again, if you capture it on a piece of toilet paper, that data on that piece of toilet paper, that's source data. The document on which you captured it is the piece of toilet paper, okay? That's what it is. So everything, wherever this information comes from, and there's a whole list that is given, but just always put it in your head that when I conduct a procedure during a clinical trial, wherever I capture that information first, that is what is going to be the source documentation, and the data is the source data. Okay, so that's it for session 5. Again, glad you're following, glad you're here. We are here for you. We are going to keep this education going on in session 6. And I look forward to seeing you there. Thank you.